Really. All right. Okay. Let's go. Yeah. So if you weren't here, you've just arrived, I guess maybe on Zoom. I'm Michael Cunliffe. I work at the Marine Biological Association in Plymouth, and I'm also an affiliate member of staff at the University of Plymouth. And what I'm going to do is maybe talk about something slightly, maybe slightly off topic, but I want to try and maybe make it on topic. Some of our recent work on the biology, so not really ecology, but the core biology of aquatic chytrids.
Maybe, yeah, to go and zoom.
So we've talked about chytrids a lot today. So I guess everybody in this room doesn't need for me to say again how important chytrids are. But if anybody on Zoom is just joining us right now, just to say chytrids are really important. They're an early diverging group of fungi. They're these I guess certainly non-dicarians, early diverging fungi, and certainly in the context of ParAqua, we're particularly interested in their role as algal parasites, but they have lots of other functions as well. One of the really maybe characteristic features of chytrids, and Silke talked about it this morning, but just to reiterate, is that they have this interesting dimorphic lifestyle. You can see this cartoon here to the right, where we've got a free swimming zoospore. The zoospore is naked, so it doesn't have a wall. It's just a membrane of flagellum. And then when that finds a host or a substrate, they metamorphose and they encyst, they make a wall. So they then produce a germ tube. The germ tube becomes a rhizoid. The rhizoid grows. And then within this structure called a sporangium, we get reproduction, so they make more zoospores. The zoospores eventually are released and the cycle starts again. And that's the basic, really basic life cycle of a chytrid. Okay, and so when we have this sort of central structure in the middle, which is our generic chytrid. So I showed this video this morning. This is our video that I love so much. I show it every opportunity that I can get. And this is basically one of these chytrids developing. So starting with the zoospore, the chytrid is growing. This is the sporangium, this is the rhizoid. Check out this swelling here called the apophysis. I want to talk a bit more about that. And then what's happening is it's making zoospores. They become more gravid, they swim around, they move around, and then at some point, that it's going to burst and the zoospores swim out and the life cycle starts again. And this is actually the model chytrid that we've been working a lot with, Rhizoplasmatium globosum gel 800. And just to make the disclaimer now, this is not an algal parasite. This is a saprotroph. So we haven't done this work with algal parasites, even though we would really like to. So, this is a cartoon made by a PhD student of mine, Davis Langdon, really trying to illustrate more widely the different functions that chytrids have. I think Silke showed it this morning. ParAqua sits right here. ParAqua is interested in chytrids that are algal parasites, Asterianella here, but they have lots of other functions as well. They kill amphibians and they degrade high molecular weight substrates like chitin and other material like lignin. And we've been particularly interested in these group of chytrids, I guess the saprotrophs, really basically because they're easier to grow. We don't have to worry about growing the host. We can grow them more easily. And that was the reason, really the reason why we work on them so much. So even though we've talked quite a lot about chytrid ecology today, this morning, and I really genuinely mean this, we know very little about the core biology of chytrids, the really fundamental biology, particularly the biology that governs these aquatic activities that chytrids have. And we think that's a real knowledge gap. I think it's a knowledge gap generally for the field of chytrids, but I think it's particularly a knowledge gap for trying to understand algal parasite, because a lot of the biology that's behind it we just don't know about. So that's something we're particularly interested in my group. So what are the mechanisms that control this really interesting chytrid life cycle? How do they develop? What biology is driving that? What core processes underpin the aquatic influence? And so that's broad, but we can be more specific about parasites. I'll talk about that at the end. And then what's also quite interesting, we haven't really talked about it in this meeting, is because of the position of the chytrids within the fungal tree, they're relatively basal, they could tell us something interesting about the evolution of fungi more generally and the evolution of eukaryotes more generally. So these are our almost three core fundamental questions that we're interested in, and we published a sort of perspective in Frontiers in Fungal Biology last year, just covering some of this. So one of the really characteristic features of chytrids that we saw earlier this afternoon with some of the staining of the chytrids infecting diatoms from Lake Stetchlin is the chytrids have a rhizoid. It's a really characteristic feature of chytrids and other organisms as well. So chytrids attach with their rhizoids, chytrids feed with their rhizoids, but actually the really basic biology of the rhizoid is really poorly understood. And so we want to know really how the chytrids form their rhizoids. Are rhizoids adaptable? Is it just random growth? Or are they somehow controlling how they're using the rhizoids? And to do this, we use our model, this rhizoclasmatum globosum, and in this study that I'm going to talk about right now, we're using confocal microscopy. So this is a way that we can generate three-dimensional microscope images of the chytrids, of the rhizoids, to try and understand what their structure is. And in fact, the thing that I just said about the importance of chytrids in terms of being early diverging, when we consider features like rhizoids, these are what we find in these early diverging fungi. And then as we progress into higher fungi, inverted commas, we see the move towards hyphae and yeast form. So, maybe chytrids can tell us something interesting about the evolution of fungi more generally. So how do we study chytrid rhizoids? To do that, this type of work really hadn't been done before, certainly not in a quantitative way. So we actually had to develop metrics to measure, to understand chytrid rhizoids. And we actually stole a lot of the ideas from neuronal biology. So biologists who study neurons, they really have spent a lot of time figuring out how to describe the shape of neuron cells. We took those principles and we applied them to chytrid rhizoids. So we used neuronal metrics to understand rhizoids, and it worked quite nicely. So, for example, in this little cartoon here, is the cover area of the rhizoid? How wide are the bifurcation angles? So between two stems of the rhizoid, what is this angle? This is the bifurcation angle. Euclidean distance, how far out are they stretching? And even this unit here that we developed, the rhizoidal growth unit. So rhizoidal growth unit is basically a unit of the rhizoid between two branches. Again, we actually, no one ever really discovers anything new anymore. Hyphal growth unit was developed by Tony Trinci back in the early 1970s to describe hyphal growth. So we applied that to rhizoid. So, I'm just summarizing the data, but what we actually show when we look at the rhizoids developing and growing is the fact that we see lateral branching dominating apical branching. So that means chytrids, or certainly Rhizoclasmatum globosum, has a tendency to produce more branches from the sides than just from the tips. And why is that important? That basically gives it a more bushy, more complex rhizoid system. And there's probably a whole load of biology that we don't yet know about that's going on in this tip that's actually stopping branching taking place. One of the things that chytrid rhizoids show is fractal properties. So we actually measured the fractal nature of the chytrid rhizoids. And what we actually see is as the chytrid rhizoid moves out, it becomes less fractal. So it's actually more complicated in the center of the rhizoid and less fractal as it moves out. So these are the first time they've been really described in chytrids, in chytrid rhizoids, but actually these are features that if we were a group of people working on higher fungal hyphae, this would all be very familiar. So that's quite interesting to us that we're seeing chytrid rhizoids behave in a similar way to higher fungi like dicarians, like Basidium mycotoa. So maybe some of the features that these fungi have are actually more ancient than they occurred in chytrid rhizoids before they occurred in Aspergillus, for example. So what are the mechanisms? What are the molecular mechanisms that control rhizoids? So again, what we did, we looked at what is known in higher fungi, like in Aspergillus, for example. What are the mechanisms that control hyphae development in those? And are they the same mechanisms that control rhizoid development in chytrids? So two things that are really important for hyphal growth. One is wall production, so glucan synthesis, and the other one is cytoskeleton production, which is mainly actin filaments. So it's been shown with higher fungi, if you inhibit wall production and if you inhibit actin production, rhizoids don't form properly. And that tells you something about how, hyphae don't form properly, and that tells you something about how hyphae form. They need the wall and they need actin. And we actually found virtually the same phenotype in the chytrid rhizoid. So when we actually poison gel 800 with two different poisons, so caspofungin inhibits glucan synthesis and cyclocalcin inhibits actin filament production, we see a completely different rhizoid morph form. So this is the actin poisoning, and this is the glucan poisoning. So again, mechanisms that are considered characteristic of higher fungi were seen in chytrid rhizoids, which is interesting about how chytrids function, also interesting in about how fungi evolved more generally. The fact that we find in inhibition of glucan synthesis having an impact on the chytrid rhizoid initially doesn't make much sense, because chytrids shouldn't have glucans. The idea is that actually glucan synthesis came into the fungi after the chytrids, so the chytrids don't have or shouldn't have glucans, or so it says in the main literature. The fact that we can see poisoning of glucans inhibiting chytrid rhizoids is quite interesting. So what we actually did was quantify beta-glucans in our chytrid, and we showed, yeah, it does actually contain beta-glucans. But also when we looked at the genome of our organism, we actually find putative glucan synthase genes. So again, these are just finding gene orthologs within the genome. We would have to do knockouts to really prove their function, but maybe glucan synthesis occurred much earlier on in the fungi than we previously thought, and it's also important for chytrid rhizoid development. And it's quite interesting. Silke was talking about this afternoon how sometimes these chitin dyes work well or not in different chytrid species. It would be interesting to know really what the composition of the chytrid cell wall actually is to make sense of this. So how adaptable are chytrid rhizoids? Are they just randomly growing, or can chytrids control their rhizoids in some sort of way that really helps them to function more in the environment? So one of the things that we did was just a really basic experiment where we basically carbon starved our chytrid. So we don't give the chytrid any exogenous carbon. All the chytrid has to do is grow on the lipid store that's been provided within its zoospore. And we saw a fundamentally different rhizoid phenotype when we starve chytrids compared to when we feed them. So again, this is gel 800. And these are the different parameters that we measured: surface area, rhizoid length, bifurcation angle. But what's maybe a bit easier to digest this time in the evening is this nice little cartoon that was produced. So this is basically the germling when it starts to produce the rhizoid. If we give them food, they produce a nice typical rhizoid structure like this. But if we carbon starve them, they produce what we're calling the searching rhizoid phenotype. So covering a much larger area, much wider bifurcation angles, longer rhizoids, and these larger rhizoidal proteins. Again, this makes sense. You're a saprotroph, you're looking for substrate. Having a rhizoid that covers a larger area increases your chances of actually finding something to feed on. Again, now we look at this, sort of, it seems obvious, but this idea that chytrids can really control their rhizoids is something that maybe we should have a think about. I'll come back with this at the end. This is a saprotroph, and Rhizoclosmatum globosum is really a well-known chitin neutralizer. So if you read Sparrow's classic text on the aquatic phycomycetes, this is the organism that is the one that you always find in chitin-rich environments. And we tried to grow gel 800 on chitin particles to see how it interacted with particles. And what we actually found was that gel 800 actually adapts its rhizoids when it interacts with a particle, with a chitin particle. When gel 800 comes in contact with chitin, it actually produces a much more branched rhizoid in response to the chitin. Again, the data are here. I've provided you the reference for the paper, but we've got a nice little cartoon showing exactly what I mean. Basically, if we grow gel 800 and we set up our experiment, so a piece of particulate carbon, this is a chitin bead, is in contact with the rhizoid, this rhizoid becomes hyperbranched. And so what we actually think is the rhizoid is able to differentiate in having a feeding phenotype and a surging phenotype. And what's really important to remember, just really reiterate, this is basically one cell. So we've got a unicellular fungus producing really quite complicated adaptive rhizoids in quite a sophisticated way. And this is something we were really excited about and something we can maybe think about in terms of how they impact parasites. So our more recent work, another thing we thought was lacking perhaps in the sort of chytrid biology world was some really detailed quantitative assessment of this chytrid life cycle. And this is a bit of a trendy sort of zeitgeist term now, is bioatlas. People would talk about bioatlas all the time. This is where you take an organism and you really characterize it to quite a high level. And so we're working with Rhizoclosmatum globosum. This is in fact, it's freshwater saprotroph. I've talked about this already, and we published this work earlier this year. So we grow our chytrid in a sort of really controlled way, so we can take snapshots of the cell cycle rather consistently. So we basically have a sample of the zoospore, the germling, the immature thallus stage. So Davis Landon, the PhD student with all this work, he really figured out how to get the culture completely timed. So 0 hours, 1.5 hours, 10 hours gives us zoospore, germling, and immature thallus. And then we couldn't coordinate the final stage. So the last sample, the mature zoosporangium stage, is actually a mix. So I'll show that a bit more. And we do two things. We do some transcriptomics, which I think most people are aware of. So we sequence all the mRNA in the sample, and this tells us which genes are being switched on and switched off. Quite straightforward. We did something quite new to us, which is SBF-SEM. So what's SBF-SEM? Which is serial block phase scanning electron microscopy. What's that? I hear you all shout out. Serial block phase SEM is really quite straightforward. We basically fix our samples in resin. The sample is then sliced with a diamond knife, and then SEM images are taken of each slice. So we're basically doing slices through the cell. We then get sent back a massive data file with thousands of TIFFs of all the slices, and then you take each slice, you identify the organelles and the structures, and then you rebuild a model of that cell. And what's quite neat with this approach is it's really quantitative. So we can actually quantify how big the nucleus is, how big the lipid globule is, and how that changes through the different stages. And we can identify everything. I just want to mention Davis, I mean, he did all this work, painstaking. I mean, I talk about it like it's easy. It really isn't. Going through all these SEM images, identifying all of these structures, and then actually stitching all of this together into these three-dimensional models. This is just an example of the zoospore, the germling, the immature thallus, and then we've got a developing zoospore inside a zoosporangium. And then Davis has to find all the structures, endomembrane, glycogen, lipids, mitochondria, nucleus, vacuoles, Golgi, and peripheral bodies. He finds all these things and puts them together. That's just reminded me something I meant to say this morning, just throwing it out there. We always think about zoospores and lipids, but zoospores also contains glycogen, and that's something we should think about when we're thinking about how they function. Anyway, I digress. And this is Davis at his when he submitted his thesis, and those of you with sharp eyes can see what the theme of the cake is that we had for when he submitted. Okay, so what do these data look like? So this is basically what we get. We get a three-dimensional model of the different life stages, and we have everything in quadruple, so 5 replicates of each. So 5 zoospores, 5 germlings, 5 immature thalli. We could only get 1 mature zoosporangium because it was so painful to analyze. So just 1 mature zoosporangium. But within that 1 mature zoosporangium, we actually then looked in detail at 5 developing zoospores. So we have these different stages. And we get data that looks like this. And I'm not going to go into it. There's just tons of data. Check out the paper if you're interested. But for example, let's endomembrane. We've got a quantification of how much endomembrane is in the zoospore, the germing stage, and the immature thallus. So we can compare these different components. So hopefully this plays over the Zoom. So this is basically the models that we make from our SEM images of the three stages where we have the replicates. So these are the averages for the zoospore, the germling, and the immature thallus. And you can see basically what they look like and then what the components are. So within this animation, things will disappear, and then you've got the color coding on the side of the different structures inside the chytrid, in the zoospore, the germling. So this is the start of the rhizoid, and then this is the apophysis, and the rhizoids are here. So we cut off the flagellum and we cut off the rhizoids. So now we've just got the Golgi, microbodies, lipids, nucleus, peripheral bodies, rumposome, striates, conclusions. These different structures that we find. Okay. So I'm just going to pick back some highlights that we found interesting, that we echoed in the paper, but maybe also some highlights that people are interested in the functioning of chytrids might care about. One thing that was really obvious, quite striking, in the zoospore that we didn't see in any of the other structures is the ribosome cluster. And so people who look at zoospores, back in the day before electron microscopy, this was referred to as the nuclear cap. This is the structure that was above the nucleus, and then it became realized that actually this is a cloud of ribosomes. So remember what ribosomes are. We talked about them this morning. Ribosomes are proteins inside cells that make other proteins. So they're the machine to make proteins. And in the zoospore, we see them enclosed in this ribosome cluster, and then as it transitions into the germling, that ribosome cluster completely dissipates. Okay. So that's the video to say exactly the same thing that I just showed you. And what we actually did, because we see this dissipation, the ribosome cluster in the zoospore into the germling, that actually makes sense because zoospores are known to be translationally and transcriptionally inactive. So what happens is the zoospore acquires mRNA maternally when it's being formed. It takes that mRNA maternally, the ribosomes are isolated in this ribosome cluster, and the zoospore basically can't transcribe RNA if it can't make protein. So it just has to swim around with all the material, all the information that was given by the parent, for want of a better phrase. It doesn't feed. And then it's only when it becomes the germling that it actually becomes metabolically active. And if you look at the transcriptome, so this is the comparison between the germling and the zoospore. So this is what's down-regulated in the zoospore compared to the germling, and this is what's upregulated in the germling compared to the zoospore. You can basically see this really basic transcriptome profile in zoospore it's just got ribosome control, ABC transporters, and a peroxisome. That's all it needs. It's got material to look after the ribosomes in this ribosome cluster. AB transporters and peroxisomes are what they would need to deal with the lipid globule. But when it transitions into the germling, we see a massive increase in all these different metabolic processes. So the organism is switched on, basically. It starts feeding, it starts synthesizing, it starts producing secondary metabolites, and now what biology really starts. Another characteristic feature of chytrids that we all know about are lipids. Lipids are really important in terms of how they function, but also the microloop, which we also talked about. And when we compare the different lipid structures in the different cell stages, so lipids in the zoospore, lipids in the germling, lipids in the thallus, they're really quite fundamentally different to each other. In the zoospore and in the germling stage, we get a single distinct lipid globule. But then when we move into the immature thallus stage, you can see all these separate lipid sort of pieces really dissipated within the cell. And what we actually think this is the genesis of new lipid. So they're actually in the immature thallus stage producing new lipid. So this is a comparison between the zoospore and the immature thallus stage. Another thing that we actually was able to do was track lipids throughout the life cycle of the chytrid, and the globule of lipid that the zoospore has never really goes away. That same globule of lipid is present all the way through the cell cycle up to the genesis of the next phase of lipids. So this is really interesting. We don't really understand why that is, why the zoospore retains an amount of lipid all through the cell cycle. We quantify how much lipid there is in the different cell stages and then the different types of lipids. So again, this is detail I don't think is super important for right now. And one of the things that Davis and I had always been fascinated with is the apophysis. And it's quite interesting how the apophysis, gel 800 has a big apophysis, and in the literature, it's, chytrids have got apophysis. Well, what's the function of the apophysis? What's it do? So we really wanted to look at that quite specifically. So what we actually find is if you look in the apophysis, it's completely packed with endomembrane. So a really high proportion of endomembrane in the apophysis compared to the sporangium. And so what we actually think is happening in this apophysis, this is basically a junction. It's a junction between the feeding rhizoids and the reproductive sporangium. And this endomembrane is how the chytrid is trafficking material that comes from the rhizoid into the cell. And what's even more exciting for us is if we actually do some live imaging. So this is live cell imaging where we actually stay in the endomembrane, and you can actually see endomembrane coming up, so material coming up through the rhizoid into the apophysis and then going into the base of the sporangium. So this is the mechanism that the chytrid uses to get material going up into the sporangium for reproduction. So this is in a saprotroph. This has got to be happening in parasites as well, exactly the same thing. What's also really cool is if you actually look at the junction between the apophysis and the sporangium, if you actually, so we're sort of looking up through the apophysis now into the sporangium, you see this little sort of pseudo septum hole in this junction between the apophysis and the sporangium that's particularly chitin rich, this little tiny hole. And so this is the way really that the chytrid cell can actually differentiate the functionality of that individual cell. This tiny little hole separates attaching and feeding that the rhizoids do with reproduction that the zoosporangium does as well. And if you think about if you are looking to learn about how hyphal fungi function, they have septa. They have between walls, between individual cells, there are small holes called septa. So we're sort of thinking, is this a early form of that differentiation that we see in higher fungal cells? So finally, the mature zoosporangium stage, we were only able to get one mature zoosporangium. It contains 82 developing zoospores. So this is the discharge plug. Not all chytrids have these. Some have the little trapdoor and different things, but this is the developing mature zoosporangium with 82 developing zoospores inside. What was more interesting is we were able to actually look at each individual zoospore, so 5 individual zoospores developing inside. So here we're comparing the developing zoospore inside the mature zoosporangium with a free swimming zoospore, so comparing these 2 different cell types. And hopefully, I think 2 things that are particularly interesting. One is the amoeboid nature of the developing zoospore. In fact, let me do that again, because that, perhaps the most exciting thing for me was the way that the flagella wraps so neatly around the developing zoospore. I wasn't expecting to find that, and that was particularly cool. But what's really important from an evolutionary perspective and the biological perspective is this amoeboid nature of the developing zoospore versus the mature swimming zoospore. Why potentially might that be important? Why should we care that the developing zoospore looks amoeboid versus the non-amoeboid zoospore stage? So what we actually think is when zoospores are developing, they actually consume maternal cytoplasm through engulfment. So they're actually engulfing that maternal cytoplasm. They take that into the developing cell, and then they use that to make the zoospore and develop and grow. Why is that such an important or interesting thing? If this is true, I mean, we think it's true, we saw it in all our replicates, but if we are seeing engulfment in the developing zoospore, this is really the first time that it's been shown in true fungi. So those basal epistosporidia that we talked about this morning, are they fungi, are they not fungi, like Rosella, they can do engulfment, but that was lost when we went into the true fungi. And so if what we're seeing is real, and we think it is, but we need to do more work, of course, then actually we're seeing engulfment in developing chytrid zoospores. And that's also really exciting from an evolutionary perspective of the fungi. So this cartoon just summarizes what we've been doing. And I guess really, we've only just scratched the surface. We're trying to get into more and more detail about chytrids, chytrids generally, chytrids in terms of saprotrophy. But I also maybe want to make the case that we need, if we really want to understand chytrids as parasites, we need to do more on the core biology. And I guess that's what I'm trying to sort of promote. And some examples of other people's work just to really make that point. This is a relatively new chytrid, isolated by Joyce Longcourt and colleagues. It was only published in 2020. It's got a genus that I'm not even going to try and pronounce this time in the evening. And this is a chytrid that infects and kills Hematococcus. So Hematococcus is a really important biotech. Green alga, it makes astaxanthin. I believe astaxanthin is worth more than gold in terms of its weight. And in Joyce's paper, which the reference is for here, Joyce actually calls them seeking rhizoids. So Joyce and colleagues say that actually these parasites, these chytrids that are looking for the hosts, are actually producing these, well, Joyce calls them seeking rhizoids, we call them searching rhizoids, but modified rhizoids that are searching for something to kill, like Hematococcus. And then one of my favorite examples that we desperately tried to work with and couldn't get anywhere is Asterianella. I think Silke showed this picture this morning from Gordon Beeks, Hilda Cantor, and others on this chytrid. And you can basically see this chytrid, it's attached to the side of Asterianella, and it's pushing its germ tube through the girdle. So, you know, this is controlled biology in action. This is not just randomly doing it. You know, what is the biological mechanism for this rhizoid? How does the rhizoid know where the girdle is? How does it find it? How does it penetrate it? I think somebody said pressure earlier this morning. I'm not so sure. I think they're secreting enzymes and almost dissolving their way in, but we need to find out. So just two examples of the work that we've done on biology of chytrids and what I think really we should be a direction that the field should be going down if we want to really understand algal parasite biology. I just want to thank everybody in my group. I'm really lucky to work with them all. Some people contributed to what I've just talked about and others didn't. And that's it. Okay.